or ideas, what you consume influences who you become. On the Mind and Matter podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a PhD in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content. Hey everyone, I want to take just a minute to tell you about a really cool health monitoring device I've been using called Lumen. It's a handheld, pocket-sized device with a sleek design. The Lumen device measures CO2 levels in your breath, which allows their technology to determine the extent to which your body is burning fats versus carbohydrates. Lumen helps improve your metabolic flexibility, your body's efficiency in shifting between using fats and carbs for energy. I use this device in the morning, or before bed, or after meals, or after workouts to track my metabolism. With just a couple weeks of use, I learned a lot about which foods were causing my body to burn mostly fat, mostly carbs, or both, as well as how long I needed to fast and how hard I needed to exercise to promote fat burning. Being able to track whether your body is burning fat versus carbs will help you figure out how your body responds to your lifestyle and whether or not your weight loss and fitness strategies are actually working. Lumen is great for anyone looking to optimize their health for either weight loss or athletic performance. Their easy-to-use app allows you to track your results together with what you're eating and how you're exercising, and it syncs with other devices like the Apple Watch. Click the link in the episode description to learn more and use the code MINDMATTER, MINDMATTER, to get 10% off your Lumen device today. That's on top of the site-wide sale they are currently running, and the Lumen device is eligible to be paid for with FSA and HSA health accounts. Check out the link in the episode description for more info.
Hi.
Can you tell everyone a little bit about where you are and what your background is and what you study as a scientist?
So I am in Bordeaux, France, and I am a neuroscientist. I work since many years, more than 25 years, on a particular system that is the endocannabinoid system. And so the idea in my background is that I am a vet of education, a veterinary doctor, but then I didn't want to do the profession. I liked the lab, and therefore I tried to do different things related to veterinary. And then when I was 30 years old, I said, okay, I need something that I really like. And I found a PhD position. So I started my PhD very late. I was already 30 years old, and it was in Germany with Dr. Beat Lutz, who started a lab and who wanted to use genetics to study learning and memory. And I proposed to him to study. This was 97, so it was a long time ago. And I proposed to him to study the endocannabinoid system in a moment in which the system CB1 receptors started to be very famous, but it was not yet very well. Not much was known already. And since then, I got in love with this system, and I say that what I say always is that it is a kind of as I wrote together with a friend that is Jaideep Bains in Canada. We wrote and Arnau Bosque Garcia in Spain. We wrote a review some time ago that was about the title. The idea was of Jaideep was the endocannabinoid system extracting specificity from ubiquity. So that means the system, the receptor, is expressed a bit everywhere. Not really everywhere, but many places. But the system, instead, the action of the system, when we will go a little bit more into details of the elements of the system, but seems to work in a very specific way. And this kind of way, really, how you extract specificity from ubiquity is a general rule of the brain somehow, because we have few elements, and with these few elements, the brain and the body are doing enormous, complicated things. And so what I say always is that our CB1 receptors basically are our way to try to penetrate a bit the complexity of the brain. So we instead of having a top-down approach in which we study a function and we go to the molecules, we try to have a bottom-up approach. We start from one molecule and we follow it into the brain where it is leading us, and to try to understand what could be general mechanisms, general properties of the brain working, of the brain and the body also.
Yeah. So, I mean, one of the things we'll talk about is the CB1 receptor and some of the other pieces of the endocannabinoid system. But conceptually, one of the things that we're going to dance around here is the CB1 receptors in the brain, it's in the body, it's in all sorts of tissues in many, many parts of the body. And one way you could start to think about it is, okay, well, the receptor is doing something in liver cells and it's doing something different in brain cells. And it's like, you could think about it sort of one system or one tissue at a time, or you could think about it holistically and ask the question, is there sort of one sort of core function that's being coordinated through the system at the organismal level? Or is this just sort of like a patchwork? It's just a type of receptor that any cell might use, but it's being used sort of in a different way in different tissues, and we'll come to that. But let's give people a sense for the core elements of the endogenous cannabinoid system, starting with the receptor, I think. What is the CB1 receptor, and what does it do at the cellular level?
So, historically, as the namesake, endocannabinoid system has been identified as the target of cannabinoids. Cannabinoids are the chemical component of the plant cannabis and marijuana, we know since many thousands of years that this plant can have effects on the body. We don't know, of course, we didn't know, of course, until, let's say, middle of last century, how the plant was doing this. Mm-hmm. And at that time, two chemists, one in the States, in the 40s, and then in Israel in the 60s, they identified the structure of these molecules that are called cannabinoids. And now we know that there are 100 compounds in the plant, that we don't know the biological function of most of them. A large majority of them are unknown. But we know that there is one component that is the responsible of what the plant is doing at the level of psychotropic level. So that it's giving the high, and this is the famous delta-9 tetrahydrocannabinol, which 25 years later, was identified as a ligand of a specific receptor in the brain. And this was cloned at the end of the 80s, and the paper was published in 1990 of a cloning of a receptor, and was called CB1, cannabinoid receptor 1. And this CB1, the fact that it was already at that time necessary to put a 1, means that they knew already somehow that there was something else. And 3 years later, I think in 93, yes, they cloned a second receptor that is called CB2, which is more expressed in the periphery. It is much more mysterious at the moment than CB1. And it is very interesting, but we work only on CB1. Mm-hmm. So once we had the receptor, CB1, and we knew that CB1 was responsive, and CB2 were responsive to the exogenous ligand. The question, of course, was why we have a receptor. It is not only to enjoy the THC somehow, but evolution or whatever will give us the receptor, because probably there are physiological functions. Mm-hmm. And to have a physiological function of a receptor, you need something that it is activating it. So very few years after the cloning of a receptor, again, the group of Raphael Mechuram in Israel, they identified endogenous ligands that are lipids that are called endocannabinoids that can activate the receptor in a similar, not identical, but in a similar way as THC does. And so at this point, it was clear that there was an endocannabinoid system in the brain that is formed by the cannabinoid receptors, CB1, CB2. There is discussion about what are the limits and what are the boundaries of a system, because if you find that there is that an endocannabinoid can activate also another receptor, is this other receptor member of endocannabinoid system? This is a debate. This is a discussion. It's more formal and semantic. For me, I would stay more on the cannabinoid receptors and the ligands that are we call so-called endocannabinoids, that are lipids, most of them, and then all the machinery that is in the enzymatic machinery that is involved in the synthesis, degradation of endocannabinoids. And all the molecules that can bind to the receptor could be considered, and activate or inhibit receptor could be considered as endocannabinoids. We have two endocannabinoids that are the most famous, 2-AG, 2-arachidonolylglycerol, and anandamide, that are both derived from arachidonic acid. But we know that there are more molecules that can activate or can, for example, endogenously allosterically regulate the activity of a receptor. So these are the elements. The minimal core of the endocannabinoid system is receptors, ligands, and machinery to synthesize and degrade the ligands.
Yeah. So there's this core to the endocannabinoid system. There's two major receptors, CB1 and CB2. There's two major endogenous cannabinoids, anandamide and 2-AG. They're both lipid-derived fatty activators of the CB receptors. And there's also the enzymatic machinery that regulates the turnover of these molecules and how long they're active. There's more to it than that, right? These molecules can bind other receptors, and there's, we can, get into the semantics of whether or not we consider those things part of the endocannabinoid system. But for our purposes today, we'll probably mostly focus on the CB1 receptor, which was the first one discovered, even though it's not the only one. It's in the brain and it's in the body. It interacts with both of the endogenous cannabinoids as well as THC. And, there's more to the story than that. Maybe we'll get into FA and some of the other enzymes, but for our purposes today, we'll be talking mostly about CB1 receptor interactions. So you mentioned that endogenous cannabinoids are lipids. Can you say a little bit more about that? How are they actually generated from fats in the body, and how do they connect to the omega-6 polyunsaturated fatty acid precursors that we think about oftentimes in the context of diet?
Yes, the production of endocannabinoids in the brain is in the cells of the body is very complex and redundant, let's say. There are a couple of biochemical pathways that have been identified, but then we realized that the biochemists that were studying these aspects realized very soon that there are different ways through which endocannabinoids can be produced or degraded. But let's say to keep it relatively simple, let's say that we have on one end we have anandamide that is produced by an enzyme that is called NAPE-PLD, I don't remember. Etanolamine.
A big long name.
Phospholipase D, which is producing finally will lead to the production of endocannabinoids. And on the other hand, you have a 2-arachidonolylglycerol that is derived from also from arachidonic acid through enzymes DAG lipase. So the deacylglycerol lipase, which means that removes a chain.
Yeah.
The deacylglycerol.
Well, I mean, one thing I want to say here for people is, on the one hand, we don't want to get buried in some of the arcane nomenclature here, but if you listen to some of these words, so we'll use a lot of abbreviations like 2-AG, 2-arachidonylglycerol, glycerol sort of like the glycerol backbone of a triglyceride,
Yes.
lipase, an enzyme that breaks down
Yeah.
lipids. They all have these big long names, but the reason that they have names like this is because of their lipid nature. And so
Exactly.
that's just sort of a nomenclature thing I'll say up front for
And
people.
what you were mentioning before, they are directly linked to the diet, type of lipids that are all the lipids that can turn somehow into the arachidonic acid cycle.
Mhm.
All these are amenable to increase endocannabinoid.
Yeah. And so
So,
ultimately,
to
this would
put it,
be omega-6 polyunsaturated fatty acids, right? So when we
yes.
when we eat things like seed oils that have linoleic acid, these can be elongated into arachidonic acid, and then that's
Mhm.
sort of like the precursor to a lot of different things, but including the endocannabinoids.
Yes, yes. Then there is a link, for example, between the endocannabinoids and prostaglandins. You
Yes.
told me, and so 2-AG is also in the pathway of catabolism of prostaglandins. So there is a relationship between inflammation and endocannabinoid levels and so on. So the thing is very complicated. Let's say that from the side of endocannabinoid is the most complicated. CB1 we can play around. We have one molecule. It's hyper complicated. But if we go to see how the lipids can be, lipids are the most difficult thing to study
Yeah.
in biology, because
Because they're
we can
ephemeral,
see
they're constantly changing and turning
they
into slightly different
change. We
versions.
cannot see them. There is
Yeah.
no way to label them.
Yeah.
The fact that you label a lipid already, you change everything. So they are very often neglected, but
Right,
not
right.
because they are not important, because they are a mess.
Yeah. I mean, a part of what you said there is they're experimentally almost intractable because being able to watch them move and track them requires you to change them. But due to the sort of fundamental chemistry at work here in lipid biology, the act of changing them to make them more experimentally amenable actually alters what they do, which
I'm not saying that it is impossible, because there are
it's just very,
many
very
people
difficult.
that are working with lipids and they
Yeah.
make fantastic work, but they are heroes because
Yeah.
they are really treating something that is, by definition, uh, uh, difficult to see, difficult
Right. Right.
to manipulate.
Mhm. Okay. So just at basic level here, when we think about the major endocannabinoids like anandamide and CB1, what happens when they activate the CB1 receptor at the cellular level? What's happening at
Okay.
that molecular cellular level?
One thing that we could say already that is important is, I mean, CB1, I don't know if we said, but CB1 is a G protein coupled receptor. G protein coupled receptor were discovered, I think, I don't know, 40, 50, maybe, I wouldn't want to say something wrong, but it's, well, a couple of Nobel Prizes given for that. And this is one of the main regulator way in which cellular processes are regulated. There is a little bit of what I was saying before, that most of the GPCRs that are studied are either receptors, physical receptors, for example, the receptors for the eyes, for the retina, for the light, or other type of physical receptors that change conformation due to physical changes on the environment, and therefore they transmit a signal. Or when they are chemical receptors, they are most of the ones that are very well known, and most of the models that we have are based on water-soluble ligand receptors. So we think about glutamate
Mhm.
metabotropic receptors, or most of the ligands, that the receptors that we consider are water-soluble. CB1, together with many others, are lipid receptors,
Mhm.
which make them already more complicated and more difficult to, and also probably following slightly different rules. One thing that we found in the lab some years ago, together with, I mean, in parallel with others, I'd say, is that if you go to Wikipedia or to another encyclopedia, whatever, and you read the definition of GPCR, at least until a few years ago, it was like that. It is written, it is a receptor, a protein that is sitting on the membrane of a cell, receives information from the external of the cell, and transmit information inside the cell. This is the let's say the lay people definition, and also the scientific definition. Many people think that GPCRs are only on the external of the cells. And this is probably true for ligands that are water soluble because they are outside. They are given by other cells. And so this is a way to make the communication.
A water-soluble ligand can't pass through the membrane on its own.
It can't pass the membrane, and they okay, you could have also from the inside of a cell some, but it's probably really a good definition for some intercellular communication. Instead, lipids, by definition, they are able to move around in the membranes, and in the cells, there are not only membranes. There is not only the plasma membrane that is the one that is surrounding, but all the organelles inside the cells are defined by circumscribed by membranes. So lipids can go everywhere. And what we found some years ago is that actually there is a subgroup of CB1 receptors that are active inside the cell more than particularly on mitochondria, which is
so you've got cannabinoid receptors on the cell membrane proper, but also inside the membrane of mitochondria and even other organelles.
But your question now, I realize that I'm going too much into a particular thing that is, for me, it's super interesting. But your question was more, what is the general signaling? What we know about CB1 signaling? CB1 is activated. Yeah, when CB1 is activated, you can have many different effects. It is a GPCR, and therefore it's linked to the activation of G proteins. There are some dogmas that are not really true, but let's say to keep in general is that normally CB1 receptors are linked to GI proteins. GI means that there are proteins that are inhibitory, which means that inhibit the production of a certain cascade, that is the cascade of cyclic AMP and pKa. Sorry for all these acronyms, but when activation of CB1 can activate many intracellular pathways, for example, different types of kinases, different types of immediate early genes can be activated directly by CB1 receptors. And so what can happen in the cells, it can be very different. It depends very much on few factors. That is, one is which cell, second is when, and third is in which conditions the system is activated.
But in the abstract, cells have all sorts of receptors on their surfaces and inside. Sometimes there's always genes being turned on and turned off in some pattern in a cell. There's always certain enzymes that have a certain level of activity, and it might be going up or going down. So when something like a cannabinoid binds to something like a CB1 receptor, it tends to have a certain pattern of effects. Certain genes might get turned up or down, certain enzyme pathways or activity levels in the cell get turned down. So when something like anandamide binds to the CB1 receptor, often, but not always, it has what we think of as an inhibitory effect on certain things that are happening inside of the cell. In general, in neurons, for example, the neuron that's activated, where anandamide activates CB1, tends to get quieter in its activity, at least temporarily.
Yeah. This is let's say a general rule. However, one thing that I'm trying to promote somehow, but I think it is also important in the study of particularly brain, but it's valid also for for the general body, also at the level, at pharmacological level. So we think one thing that so in my career, in these last 25 years, more or less, what we used is models in which we were able to delete CB1 receptor in different cell types.
Mm-hmm.
So to try to disentangle somehow how CB1 could modulate the activity of this or that cell. So one of the conclusion to which we arrived, it sounds a little bit trivial, but it is not, is that the activity of CB1 receptors is not intrinsic.
Mm-hmm.
So you cannot say that CB1 receptors has this activity, but it is an emerging pattern, an emerging activity that depends on where the receptor is, in which condition the cell is, when the endocannabinoids are released, when they are degraded, how long is the activation. So it is, you can have the same thing, one thing, and exactly the contrary.
Mm-hmm.
I make you another examples. Very often we think about the endocannabinoid system and we say, okay, we block the system, we delete the receptor, we do something, and we have this phenotype. Good. That means that the endocannabinoid system is probably important for this function. Okay? Then we go and we inject THC. And we see that the THC is sometimes doing exactly like the absence of a receptor.
Right, right. So this would be to link this to something that some people have experienced. Right, when we think about THC and the effects of THC in people, you can often get contradictory or opposite effects in different people, or even within the same person at different times, right? Sometimes it wakes you up, sometimes it makes you sleepy, sometimes you get hungry, sometimes you don't.
Absolutely. And so and one explanation that we have for that is that the endocannabinoid system is very specific in times and space, time and space. So only when and where, because we spoke about the production of endocannabinoids, but we didn't speak about the degradation of endocannabinoids. It's a very efficient machinery, very efficient. So as soon as the endocannabinoids are going up, there is immediately some enzyme that is trying to kill them.
Yeah, yeah.
And because the system is really limited in time and space. In the moment in which there is also evidence for tonic activation of a receptor.
There's some tone, but in general, this is a highly spatial,
in general,
temporally
it's
precise.
on-demand activation.
Yeah.
Instead, if you go with an agonist, a THC, the time and the space are completely lost.
Yeah.
The selectivity, because the ligand will go everywhere and will stay. Only the only limit will be the time of kinetic of agonist, or the receptor that is recycled and is inhibited, and so on and so on. But so sometimes it's really the moment that is determining the function, and not the activation or non-activation of a receptor.
So in contrast to something like THC, or even just another drug we might think about, right? If you ingest THC, it might take hours of time for the THC to be fully metabolized by the body, and it's going to go everywhere all at once. But the endogenous cannabinoids, these lipid-based molecules, they're made very specifically at certain times and very specifically at certain places. One synapse will make some anandamide, and then it will be immediately degraded. And so this
is what we think. And talking about what you were saying, that what everyone experiences, who experiences the use of cannabis, can describe, or people can describe, that someone has one effect, someone has another effect. One day you have an effect, the next day you have another effect. This we see also in animals. The effects of cannabinoids are almost always, we say, pharmacologically, we say biphasic. That means that you have a dose that is decreasing, having an effect, for example, decreasing anxiety in animals. in animals. You make the experiment. You take your plasmase. It's a test of memory, or a test of anxiety for mice, and you inject a very low dose of THC, and you see that the animals are bolder. They are less worried about an anxiogenic environment. Then you increase a bit the dose, and you find that the animals are absolutely normal, like the controls. No effect anymore. Then you increase the dose, and you see the mice that are absolutely frozen in a corner, and they don't want to move where there is any slight idea of a potential danger. So they are very anxious.
Mm-hmm.
So you have completely opposite effects, and we together with others, we found that actually this on anxiety we didn't do. It was my former supervisor, Beat Lutz, and he found that different populations in the brain of CB1 receptor on different cell types are responsible for this.
Mm-hmm.
So we will come to speak about this, but it looks that this the fact that CB1 receptors is in different cell types, it is used by the body and the brain to give signals, but then to say, okay, when it is a bit too much, let's start to activate the other one that is doing the contrary.
Yeah.
And this we will come probably talking about what you wanted to discuss more mainly.
Yeah. I mean, so both at the cellular level, and I think as we'll see at the organismal level, we come to the idea of compensation or homeostatic regulation. A lot of things in biology are based on negative feedback loops that keep things in a certain range. You don't want your blood glucose to get too high or too low. You don't want neural activity levels to be too high or too low, right? There's this sort of Goldilocks regulation that you see in biology. And we think about that, the term for that is homeostasis. And the CB1 receptor system seems to be very closely aligned with this idea of homeostatic regulation.
Yeah. Yeah. Okay. Here we are coming to talk a little bit about this idea of what homeostasis is,
Mm-hmm.
at least, and there is this, in my opinion, it's valid for for all functions of the body. There is one domain in which this is very clear. This is it's very clear. This is the energy intake and food intake. Okay, I have to say immediately that the idea, the original idea, is by Pier Vincenzo Piazza, that is the former director of his institute, and with whom we had a lot of discussion. And when I was talking to him about the endocannabinoid system, he came really to this thing. But come on, guys, this is really he works. He used to work mainly on drug addiction, on cocaine, on studying that, but he had these ideas about how the balance of the body and the brain can be altered. For example, drug addiction is a typical allostatic state in which another homeostasis is what you need.
Mm-hmm.
So
mm-hmm.
you need the drug to be normal.
Yeah, basically you start ingesting the drug so much that you get to a new stable state of being.
Exactly. So this is allostasis. It's called like that. But when I talked with him about the endocannabinoid system, and we were discussing together, and he came with this fantastic idea that is so to make it as simple as possible. What is so homeostasis is a general word that is valid for everything. I mean, we can say homeostasis is. We were laughing at some point also because PRV told me sometimes I read that there are physiological systems that are defined as non-homeostatic, which doesn't make much sense, because something that is non-homeostatic should be selected by evolution immediately, or as fast as possible, to be eliminated. So everything what is remaining has probably some homeostatic functions. Then what is changing is the timing when you do something for which reason, and this we can start to analyze a little bit. Starting, for example, from the food, from the food and then energy balance. So the idea of Pierre was simply like that. We have two forces, we have two conditions that are determining our homeostasis, which is our fitting, our physiology. It's not his idea, it's described, but to you, what his idea was to use this particularly good terms that are endostatic and exostatic. So if I ask you why we eat, the first answer will be because we need energy. Okay? Because otherwise we die. That's very simple. However, this does not explain why we eat more than what we need.
Right. We finish eating a big dinner and then we have dessert. Why?
And then we have
Even
dessert,
though we're full.
and then if they invite us again, we keep on, and so on and so on.
Yeah.
And
so at an intuitive level, what we're talking about here is, what everyone might recognize as, we eat for our stomach sometimes because we feel hungry because we haven't eaten, but very often we eat for our tongue or for the pleasure of engaging in the sensory experience.
Exactly. And so the fact that the term of pleasure then is something that is coming later, because what is the point? So what we can define as endostatic is the behaviors and biochemical physiological processes that are leading to a compensation of an immediate need.
Yep.
So I need something, my body sees that the level is going down. I have sensors of glucose, sensors of nutrients in the brain and in the body that are telling me, look, we are getting off. So I need to compensate this.
And I think a key here is that system is very much in the here and now. Things are going down now, and so we need to replenish them now. We're not really thinking about the deep past or the future. Yeah.
Actually, don't say the word thinking, because even the other one is not thinking.
Right. Yeah.
But
it's an immediate need.
It's an immediate need. And in the moment, the needs of now are compensated by an endostatic system that is called homeostatic. In some papers, it's called homeostatic. What you said before, compensation makes you think immediately to homeostasis. But then you say, what is the other one? It's non-homeostatic, which doesn't make any sense. So, and one characteristic of endostatic processes are that they are compensated by definition. As soon as the level is filled, you stop.
Yep.
You don't have, the reason why you were doing that thing, or that your liver was doing that thing, or that it can be anything, right? Is not there anymore.
Yep.
So
it stops. It's like a thermostat in your home. The temperature goes down, and so then the heat turns on, and then once it gets back to the set point, it turns off. Exactly.
And this would be perfect in a perfect world.
In a stable environment.
In a stable environment. In an environment where you have always the right amount of food available, you have always the right amount of different nutrients that are necessary for your life. The food is variated.
Yeah, but if there's seasons and you're a bipedal ape and you're exploring new territory and there's a famine.
Exactly. The problem is that the environment is a little bit bizarre. It's changing continuously. Even in the same generation, even in the same life of individuals, you could have periods in which you have abundance and periods in which you have less abundance. And this has been always, until when we did not invent the fridge, basically. The food, we needed to make much more work to get the food, and we need to have supply.
So the stable food environment is a present day anomaly from an
I
evolutionary
know.
perspective. For most of
Exactly.
human history, we lived in a much
But
less stable environment.
let's stay in what is normal, that is the less stable environment. In a less stable environment, you need, no, let's imagine that you are endostatic and therefore you eat only what you need. I am a little bit, you are very thin, and I instead tendency to become a little bit round because I am a little bit less endostatic than you. I am a bit more exostatic.
Yeah.
What does it mean, exostatic? Exostatic means that the stimulus to make me eat
Mm-hmm.
or to have some processes in my body is not inside me, is outside me.
Mm-hmm. You're driven by food stimuli that you see in your environment rather than the internal
It
need
is
or
not
hunger for something.
it is not my body telling me eat. It is the food that is telling me eat me.
Mm-hmm.
Okay? And what is the reason of that? I mean, one thing, if you think now, it is this something that is making you eat more than what you need is pathological, is
Mm-hmm.
making us sick and
Yeah.
fat and diabetic and so on and so on.
But it's so
In,
common
in
for people, it's so common for people to have that exostatic way of doing things, which from an evolutionary perspective has to make you think there must actually be a good reason that tendency is so
Mhm.
common. So if we think back
Exactly.
to when things were unstable, I'll let you finish the thought here.
Exactly. And this exostatic thing is opposite to the endostatic, is a way that evolution found to respond not at present needs, but future needs.
Right.
So the
Prepare
idea
for
is
the
that,
future famine by feasting now when the food is
yeah,
present.
exactly. If I am more exostatic than you, we find some food, you eat what you need, I eat more, then we stay 3 weeks without finding food, you die, and I survive because
Mm-hmm.
I have some reserve.
Mm-hmm.
Okay? That's the description. And but there is an intrinsic problem in this. Where is the motivation to do something, to spend energies now for something that might be useful in the future?
Mm-hmm.
And evolution does not make you spend, is conservative, doesn't want you to spend energy for nothing, right? Of course. Uh, uh, um, but what happens is that as you will survive if you do that, you need a motivation. And the motivation is given by the rewarding system that is given by the food itself.
Mm-hmm. So this is where we talk
So
about the palatability of the food.
palatability of the food, but also a lot of biochemical activities that are leading to an accumulation of energy. So one thing is the food, that is the palatability, the food that is telling me, eat me, and it has more, it has characteristics that are reminding me something, or that they are good, that they are combining different nutrients. For example, sugar and lipids is much more palatable than sugar alone and lipids alone,
Right, right, right.
because this makes a
Yeah.
combination.
So what we're
And
doing here is we're actually getting at, think about it for a second. It's not a coincidence that the most palatable foods, the foods that we tend to think of as yummy, have things like sugar and fat in abundance.
yeah,
Nobody imagines eating a bunch of celery
mhm.
to prepare for the future. It's
Yeah,
just
yeah,
those
yeah, yeah,
foods that are the best at getting you to literally put on pounds, which corresponds to energy you can use in the future.
yeah, 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 yeah. And this is absolutely like that. And what is the feature? What is the problem with a feature of this system, that it is like a I don't know the term in English, but it's like a system that is self-perpetuating. So whereas the endostatic systems are compensated, so
Mm-hmm.
they stop, they have a mechanism to stop. The exostatic systems are the tendency to be
Continue.
to continue to not to be compensated
Yeah.
because they're based the evolutionary reason why they exist is not to stop.
Right. Because the environment would force you to stop at some point in a time of famine, say.
Exactly.
Yeah.
And therefore and we have and the very great idea of Pierre was yes, the endocannabinoid system really seems a system that is uh, uh, um, cut to do this. So this
Hmm.
is what you mentioned before is not just a piece of different pieces that are different doing different things. This
Yeah.
could be a general idea about the system that is a system that is made to make us do a little bit more of what we are doing.
Yes. And if the CB1 receptor system is an exostatic system and its purpose, the reason it evolved, is to get an animal to do things and to coordinate stuff changing across tissues in order to store energy now to not die in the future when food is unavailable, then we should see a sort of a pattern in terms of the types of cellular and tissue level changes that happen from CB1 receptor activation across the body.
Yeah.
We should see a certain pattern of, say, change in the liver or with glucose homeostasis and things like this, in addition to the brain-based things like the hunger and the palatability related stuff that I think we'll talk
Yeah,
about.
for example, what we can say is so if we think about the CB1 receptor in as an exostatic system, and we so no, let's like that. Let's think what an exostatic system should do.
Mm-hmm.
When an exostatic system should, in terms of food intake and energy accumulation, should increase the amount of food that you eat, increase the amount of food that you absorb, decrease the increase the metabolism of glucose, glucose utilization, and lipid accumulation and production of lipids. All the molecules in the body
Mm-hmm.
that are made to conserve energy.
Yep.
Okay. And this is exactly what happens. The endocannabinoid system is a multi-factorial system that for which most of the actions lead to accumulation of energy.
Yeah. Can we go through some specific examples for people, because the endocannabinoid system is most famous for its nervous system components.
Mhm.
What happens, say, in the liver when CB1 receptors are activated?
But in the liver, for example, this was a discovery of my colleague and friend Daniela Cota. But there is no, sorry, no, liver, no, liver. She was working on adipocyte. Sorry, I mix up. In the liver, what happens is that CB1 is, for example, contributing to lipogenesis
I see.
in the liver.
So fat
And
synthesis by the liver.
for example, if you give high-fat diet to a mouse without CB1 in the liver or without CB1 in general, the accumulation of lipids in the fat liver will be much less strong than in control
I see.
animals.
So CB1 receptor activation in the liver is directly related to lipogenesis, how much fat is actually produced in response to fat intake in the liver.
And the same thing that I was mixing up is the discovery of Daniela, that this happens in a different way, but at the level of adipocytes, where CB1 receptors contribute to the accumulation of lipids.
I see. So they're regulating how much fat is actually accumulating in fat cells.
Exactly.
Yeah. And one of the things you pointed out in this paper is the observation that you see bona fide CB1 receptors in all vertebrates, which is all animals that have adipose tissue. And so you sort of see the system emerge in the animals that are capable of accumulating energy for the future.
Yeah, this was actually a smart request of a reviewer. We are not nor me nor Daniela nor Pierre. We are evolutionists. But of course, all the story of the paper was based on some evolution knowledge or ideas, evolutionary ideas. But the reviewer actually very smartly told me, asked us, sorry guys, but you didn't mention when CB1 is appearing. And so I told myself, I don't know. It's 27, 25 years I work on the thing. And so I called a friend that has been working on evolution of CB1 receptors. And I said, but sorry, what is the first? And he told me, it's reptiles. And it is just an observation. But I mean, coincidence is coincidence. Reptiles are among the first species that are developing unadipose tissue to accumulate energy. This is so could be that there is some relationship, but in the moment in which animals started to develop this need to accumulate, then the system appeared or became stronger and became an important element of the physiology.
Well, I think another thing I want to ask you about here is if an exostatic system, so eating for future need, driven by food stimulating your environment, exostasis. If the CB1 receptor system is exostatic, we would also expect to see certain patterns with respect to its effect, or the effect of stimulating CB1 or knocking out CB1, say, on cue-induced feeding rather than baseline feeding. So does CB1 seem to be involved in cue-induced or food-induced feeding in particular?
Yes, it is important for the palatability of the food and so on. But of course, you have to think about something that there is the idea when you make a theory, when you separate things and distinguish and isn't say, okay, there is this thing and this thing. It's never like that.
Right.
They overlap. So it is extremely rewarding to eat when you are hungry.
Right.
So it's not that the endostatic has nothing to do with the reward, or
they're intertwined.
Exostatic has nothing to do with the immediate. It is that the two systems, for example, we published a paper some years ago in which we found that CB1 in a certain circuit that is going from the olfactory cortex to the olfactory bulb controls how odors are perceived, particularly food odors. So if animal is food deprived, then the endocannabinoids increase, and this will make them smell the odors much stronger and much more attractive.
So basically, what you're saying is,
for example, I don't know if you ever happened to you. I hate when you pass close to a fast food, whatever. Very often the smell is disgusting. When you can like maybe the food inside, but the smell is not good. Actually, they made in Milan, in Italy, they made close McDonald's just for the smell. But if you are hungry and you have a smell of a fast food, you think that it is the best smell of a food.
So that's an experience that everyone's familiar with. When you're hungry, the sensations just hit different. They're much more appealing. What it sounds like what you're saying is what people have shown experimentally in rodents is that literally in a hungry state, a food deprived state, the endocannabinoid production goes up in the olfactory system in a way that makes the animal, that basically enhances olfactory perception.
Yes, that's one example. For example, in which the two things overlap. It's the fasting, it's the lack of energy that makes the system stronger. So it's making maybe a little bit more exostatic, even to the normal food intake. Could be something like that. For the food, this is very clear. I think the idea that there is an endostatic and exostatic component, and because the two components are very close to each other. So now I'm talking about things that are not that I didn't write. But Pierre mentioned them, and he developed a very good idea. Pierre Piazza in a book that he wrote that is called Homo Biologicus. The problem is that it is only in French and in Italian because he's Italian, and we are still waiting that there is a translation in English. But if somebody can read these languages, it's a suggestion.
Well, but let's give people more a sense of what's happening in other tissue systems. So if the CB1 receptor system has as a core component of it this exostatic function, so we said that the liver is engaged in lipogenesis, and CB1 seems to be involved in that. What about things like just nutrient utilization and absorption through the GI tract? Wouldn't we expect an exostatic system to enhance nutrient absorption?
Yeah. It has been shown that, for example, one thing that the cannabinoid system does is to reduce the motility of the intestinal tract. Like that, food has more time to be absorbed.
So gut motility goes down, and so that allows you more time to actually absorb what's going through the gut.
Exactly. But there are many examples like that. The glucose utilization by the muscle is is reduced because like that can go to the production of energy storage and not to the consumption.
Saving for the future instead of using it right now.
Exactly. So it's really it works in the case of energy metabolism is pretty much clear. I mean, it is a system made for us to accumulate to respond to future needs.
Yeah, and so this would, I mean, when you start to think of things this way, it's much more natural to think about why so many people struggle with things like obesity and weight loss today, because the environment, from a caloric perspective, it's not variable. We always have more than enough food around, and people really struggle not to eat, not to be driven by external stimuli, i.e. in an exostatic fashion.
Yeah, absolutely. I make you also other functions that could seem not too much related to energy intake or energy accumulation could be related to the same thing. Is CB1, let's say physiologically, all the evidence that we have so much for now is that the system is anxiolytic. So it is a system that when you have a flash of endocannabinoids, you calm down. Okay, that's pretty much evident. When we block the system, so there was just to make you an example. Until 2009, 2008, 2009, there was a CB1 antagonist, a blocker that was on the market for 2-3 years.
Yeah.
For against diabetes, against obesity.
Yeah. This is rimonabant?
Rimonabant, yes. And it worked, but then the drug was withdrawn from the market because it had psychiatric side effects. So probably the company was not careful enough in selecting the type of patients, but if one was prone to anxiety, rimonabant was really increasing it very much. So the system is anxiolytic. How this relates with this accumulation of food and accumulation of energy, with increase of food intake, accumulation of energy. It is that if you are anxious, you don't go out to look for food. So
right, you withdraw, you stay safe in a little corner.
You stay safe, but you don't fall. So you have to win your anxiety to go out. The same thing is for pain. The endocannabinoid system is a powerful analgesic system. Yeah. It's contributing to reduce pain sensations. It's the same thing. If you are on the search of food, if you feel less pain, you will have more chances to look for more food.
Yeah. To go out into the
Because
world and
you
take
feel less
risks
pain.
to find
If
food,
you
you
have a
need
lot
to
of
be.
endocannabinoids, you don't feel pain, and
Yeah.
you keep on searching, you keep on exposing yourself to dangers, and so on and so on.
Mhm.
These are some examples of this, which, if you think, is a kind of danger. In it, exostatic systems are uncontrolled,
Yes.
as we said before.
They're
They are
self-perpetuating.
self-perpetuating and expose you to dangers.
Hmm.
I mean, if you are constantly under the impact of endocannabinoid system, at some point somebody will eat you because you are going out of your nest too much. You
Hmm.
should stay a little bit more put,
Hmm.
okay? Because there is a dangerous
Hmm.
environment out there.
I want to ask you, so with this self-perpetuating nature of an exostatic system in mind, I want to go back to the idea that endocannabinoids themselves are lipid derived molecules that come from omega-6 polyunsaturated fatty acids. Is there a connection here? Because if you're in an environment where food is suddenly plentiful, and the whole purpose of having exostatic regulation is to keep taking advantage of that food as long as it's around, because at some point it may not be, then one way to bake that into the system is to make the molecular mediator of that signal, this anandamide, say, or one of the cannabinoids, to be derived from a calorically dense food source itself.
Absolutely,
So,
absolutely.
yeah.
It is probably this the reason why evolution picked up the endocannabinoid system for that, because I mean, I say absolutely because I agree with the theory, with the idea. I have no proof that it is like that,
Mm-hmm.
but makes a lot of sense.
Yeah.
What you say makes a lot of sense. That it is a system that is not only making you accumulate more lipids, is also using these lipids to make more.
Right, right.
You accumulating more lipids.
Do we have direct evidence in experimental animals that increasing the amount or the proportion of omega-6 fatty acids in the diet
Yes.
leads to a higher overall level of CB1 receptor signaling via
Yes,
increased
there are
endocannabinoid
papers
production?
from there are several papers, and now comes to my mind two or three papers that Sophie Laillé and Olivier Manzoni in here in Bordeaux and in Marseille, in France, always. They clearly showed that omega-6 diets are leading to an increase of endocannabinoids, and that increase the behaviors induced by endocannabinoids, that increase the electrophysiological effects of endocannabinoids. Yes,
I see.
it's
So,
much, but pretty much clear. Yes.
okay, so you just increase the omega-6 intake availability in an
Not
animal's
the
diet.
only way to increase endocannabinoids, of
Yep.
course.
Yep.
It's not the only way, but this is
Yeah.
a clear
Yeah.
one.
There's a direct link here. If you increase omega-6 intake in animals, they get more endocannabinoids. You see more activity, whether it's in the brain or elsewhere, you see things move in this direction.
I tell you more. We have now evidence. I don't tell you because it's not yet published the details, but there are specific populations that are specialized of cells of astrocytes in the brain that are specialized in the metabolism of certain lipids that are very excellent producer of endocannabinoids.
Hmm. So
So that
they
really
detect
that there is a
specific
kind of
lipids.
specific subpopulation of cells that are doing that job.
Interesting.
Yeah, this is
Hmm. So it's not so in your view, so as someone with this research perspective, it's not only is food, not only are calories generally abundant today, but the omega-6 polyunsaturated fatty acids are disproportionately abundant in the modern
Mhm.
world.
Yeah.
Do you think this is directly related to why we have an obesity epidemic and things like that?
As I said before, I don't have a proof. I don't know I should maybe ask Daniela, but I don't think that there is any human study that is really, yeah, there are human studies that are many human studies that are high fat diets, obesity is increasing endocannabinoids, all this, but there is a direct link This I don't know, but it makes so much sense.
Right.
What we know is that if you eat too fat, your endocannabinoid system will be potentiated, and probably you will eat more fat.
Mm-hmm.
So it is a self-perpetuating.
Yeah. And again, it makes so much
And
sense.
what are the molecular identities of this? There is some evidence, but not. I actually don't know the details. But
Yeah. But it makes so much sense when you step back and you take this evolutionary perspective and you think about time and the fact that for most of human history, most people were not in a stable food environment. Things were seasonal, things were unpredictable. There were times of feast and famine. So it made perfect sense that there would be a survival advantage to any organism that had baked into its biology the tendency to keep eating as long as food was around, because it was inevitable that at some point it wouldn't be anymore. And if you didn't stock up and store with the future in mind, so to speak, then you would starve at a time of less abundance.
Yeah. And there was one thing that I wanted to add to what we were saying before, that these exostatic systems we were mentioning a little bit that expose you to dangers.
Mm-hmm.
For example, the one of eating too much.
Mm-hmm.
It's a danger. So what we found, and we and other people found, is that what we were saying before, that very often, and this is probably the at the basis of this biphasic effects of cannabinoids, to do one thing or the contrary, it is a kind of safety system, safety mechanism, for which when the system is too much activated, there is a way to produce and to activate. For example, I can make you two examples of this. We found out that CB1 on excitatory neurons in the brain is promoting food intake.
Mm-hmm.
CB1 in GABAergic neurons, in inhibitory neurons, instead is inhibiting food intake.
Hmm.
And if you do a dose response of an agonist of a THC, you will find that the increasing food intake, the munches of THC,
Mm-hmm.
are due to CB1 on glutamatergic cells.
Mm.
But when you increase the dose,
Yep.
even animals will stop eating.
Right.
And this is due to CB1 on the GABAergic cells.
Yeah. So
and this is also true physiologically. When there is too much endocannabinoids that are going, and what is interesting is that CB1 on glutamatergic cells in excitatory neurons is much more sensitive to endocannabinoids and to cannabinoids than CB1 on GABAergic cells.
So the initial release on the lower the dose or medium dose, you get the excitatory effect. If you keep going, you get the inhibitory effect. The whole system is well set up to sort of have this biphasic up or down, back or forth.
And another very, very, very interesting thing that again was something that I contributed with Pierre Piazza, but this is his discovery, is that there is a neurosteroid, a steroid that is produced in the brain that is called pregnenolone.
Pregnenolone, yeah.
Pregnenolone, that is a natural signal-specific inhibitor of CB1 receptors.
Yeah, this is important because most people don't know this about pregnenolone. So for those that don't know anything about pregnenolone, this is basically, it comes after cholesterol and before the other steroid hormones. We think of it as a steroid hormone precursor, but what Giovanni's talking about right now is it's also got a signaling function. It's an inhibitor, essentially, of the CB1 receptor.
Yes. So pregnenolone has been always considered the inactive precursors of steroids.
Mm-hmm.
To make it very simple, what you said, you have cholesterol, which happens actually in the mitochondria, which is also interesting. Cholesterol, and then you have production. The cholesterol becomes pregnenolone, and then there are a bunch of complicated enzymes that are attacking the pregnenolone at different sites and changing the residuals and so on and so on, the residues and so on and so on, and making all the steroid like estrogen, testosterone, glucocorticoids, all the steroid molecules that we know. Pregnenolone has been always considered as inactive precursor of all these that are the big family of steroids.
People have thought about it as just merely an intermediate.
Yeah, actually, the story is interesting. It's funny because it was before I was coming here to Bordeaux, and Pierre, who was the director of the institute already, he had working on drug addiction. He had this idea that maybe steroids are important for drug addiction. So they developed this. I'm talking to you about this about 20 years ago. So there was not so much methods to measure steroids because, again, they are lipids. They are difficult. They are so we could measure only few of them at a time. We couldn't measure the whole family or a large number. So he took a technician and invested some money, and we started to work on a way to have a measurement of steroids in the brain, having a bunch of them, 6, 7, 8. I don't remember how many they can measure. So, and then I was going to come here. I was in Germany at the time, and he called me. And he said, look, I did the experiment. The first experiment is to take all the drugs of abuse, THC, cocaine, morphine, heroin, and inject rats and measure in the brain this damn neurosteroids.
Yeah.
And he told me, look, when we inject cocaine, we have some changes of 20%, 100% increase or decrease a little bit, but things that normally you would say, oh, this is fantastic, this is a great result. But when you inject THC, you increase 14,000%.
Pregnenolone goes up a lot after
14
THC.
times.
Yeah.
14 times the pregnenolone.
Which is huge.
Which is huge. It is really actually an endocrine increase.
Yeah, yeah.
And so we were, when I came here, I sent him some mice because we had to check if it was CB1 dependent. They didn't have anything, any tool for CB1. I had the CB1 knockout. So yes, it was CB1 dependent. So the injection of THC activates CB1, which at low doses doesn't do anything. When you increase the dose, boom, you have this increase of pregnenolone.
So pregnenolone is being synthesized in neurons, presumably from mitochondria, in response to CB1 receptor activation?
Yes, presumably in neurons. We are trying to understand now where is the site of.
But it's a huge effect.
It's a huge effect, but then the problem started for us, particularly for Pierre, who was leading the project, and to say why. So what is going on? What is the reason? What is this pregnenolone doing? And my suggestion was sincerely always working maybe pregnenolone will mediate some of the effects of THC.
Yeah.
So you inject THC, you increase pregnenolone. When you go over a certain dose, you increase pregnenolone. Pregnenolone does something that makes animals, I don't know, be anxious or less, whatever. All these things I told you that they are depending on high doses of cannabinoids. And so they started to make the experiments, and the results were not fitting. It was not like that. I mean, when we, at some point, once I remember discussing, Pierre said, and if it was the contrary? I said, contrary what? That it is inhibiting CB1. We went to the data and said, yes, here's fitting, here's fitting, here's fitting. So we had a preconception that and when we realized this, we said, wow, this is a natural inhibitor.
Yeah. So pharmacologically, what is pregnenolone doing?
is binding to a specific but when the paper was built up with a lot of studies, biochemistry and so on. So now we know where is the binding site of pregnenolone, which is completely different from the binding site of endocannabinoids or cannabinoids. So it's not an inhibitor, it's not an antagonist, but impedes certain conformational changes of a receptor and therefore impedes certain signaling that are, for example, the production of MAP kinase, which is one thing. And another thing that that pregnenolone blocks very well are the effects of cannabinoids on mitochondrial activity.
So it's acting as a negative allosteric modulator. It's limiting the activation potential,
not inhibiting everything.
Right, not inhibiting everything, but just
for example,
limiting.
Cyclic AMP, the cyclic AMP doesn't seem to change.
So it's sort of focusing what can come through this channel,
and it looks like that it is removing bad effects. It's a safety system.
Yeah.
Looks like that for example, we made a paper showing that when THC is given and we can have psychotic-like effects in mice, observed. These are blocked by pregnenolone.
Yeah. So, and this would imply, I mean, one thing that I think is implied by this is that you would expect to have more severe side effects in response to THC administration at a given dose for someone who had impaired steroid hormone synthesis.
Yeah. This is something that we would like to explore, to measure, for example, in people that are more or less susceptible to
well, what's
effect of THC.
What's interesting and potentially disturbing about that is there are plenty of pharmaceuticals out there that impair this type of thing. So it's plausible that there's people out there that do have impaired steroid hormone synthesis and then could be subject to this.
Yes, but I mean, trying to understand what happens with the steroid hormones it's complicated, because and this is the reason, for example, why pregnenolone in reality cannot be used as a drug, because pregnenolone first is very badly absorbed, because the first idea was, I mean, we have something that can block the effect of THC.
The negative effects, apparently.
Mostly the negative effects. And so you cannot use it as a drug because it is very badly absorbed. And then, of course, it's still a precursor of all steroids.
Right.
So if you take a lot of pregnenolone, then maybe you get too many estrogens, or maybe
it's poorly absorbed, but also probably it's highly sensitive to the dose.
So you are playing with fire because you don't know what happens. Okay. Yeah. So the idea, there was a company that was made to do that is called Elis Pharma, and the idea was to modify pregnenolone in a way to impede the modifications, the enzymatic transformation into other steroids.
Yeah.
And these, for some compounds, improved the absorption also. So basically now there are compounds that are in clinical trials trying to fight against
it's essentially a modified pregnenolone that's more easily absorbed, but also can't be used to create other steroid hormones.
Exactly, cannot
right.
Cannot be used to exactly. This is so this is an interesting example. I say always of pharmaceutical research that is driven by research.
Right. Well, and
by basic research, because this is something that in the brain for neurological disorders we have something, but for neuropsychiatric disorders there is almost nothing that came to research from the research. to the clinics. All what we use, antidepressants, blah, 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 they were found 50 years ago by chance. And having this thing that is coming directly from the molecular mechanism to the use to this to stimulate the safety system of the brain against excessive cannabinoid activation. It's pretty much exciting at the moment.
It's the pattern here, so activation of CB1 receptors leads to production of a negative allosteric modulator of the CB1 receptor, that's pregnenolone. The pattern there reminds me of like feed-forward inhibition that you see in neurons or in transcriptional networks, where you have this limiting motif built into the system, where if an excitatory neuron excites some other neuron, it's also exciting in parallel an inhibitory neuron that limits that activity and prevents essentially epileptic activity from getting out of hand. You see that at the level of transcriptional regulation, right? Transcription factor will activate some genes, including a repressor of itself, and that prevents things from exploding or getting out of hand, so to speak.
Yeah, absolutely. It is a similar thing, but it is a molecular process, and is not one cell to the other or whatever. It really looks like that it is the system itself that it is using other molecules. So pregnenolone should be considered as an endocannabinoid, because it is binding to the receptor and changing the activity of the receptor. And one thing that is important is that it is not blocking all the effects of the receptor.
Yeah, it's just limiting some of the effects.
Certain stay, probably because it is selected by evolution, probably the ones that are not harming the body. So this is as I said, it's really exciting, and yeah.
I want to share my screen real quick, and because you have this really nice figure in the paper that we've been discussing, and this kind of puts in one little cartoon here this idea of the CB1 receptor system being an exostatic system, that it's coordinating action centered around increased energy storage. And what you've laid out here are the effects of CB1 activation in different tissues. And we've talked about some of these things like the effects in the liver and so forth. But, I don't know if you want to comment on particulars here, but when you lay it all out like this, it really does kind of paint this interesting picture where all of the sensory perceptual changes, all of the nutrient acquisition and glucose homeostasis changes, all of these things really do seem to be geared towards getting the whole animal focused on energy accumulation now.
Yeah, absolutely. This was a very nice figure, and now there is even more. This is a paper of 10 years ago. We have to remind, and there is even more on this, and I don't know if I should go through all the elements, but all the points of this circle are indicating more increase of energy storage. So
more nutrient absorption in the gut, more insulin secretion by the pancreas,
more fat. For example, CB1 in this I forgot. CB1 in the gastrointestinal tract has been suggested to promote fat preference and fat intake. So if you activate CB1 in your stomach, you will feel more like eating fat, which means that you will accumulate more energy. And on the pancreas, to increase insulin secretion, to decrease the glucose utilization, and to allow, for example, for decrease oxidative metabolism. For example, this effect on mitochondria is also probably contributing to that, because we found some years ago that CB1 in mitochondria is present also in the adipose tissue, and there can decrease the activity of mitochondria, and therefore, if you decrease the activity of mitochondria, you decrease the oxidative metabolism, and you accumulate more energy. You use
stop
you are
burning
less
so much
efficient
fat and start storing it.
using. So basically, the most efficient way to use energy is to use mitochondria. If you use less mitochondria, you will be less efficient in using energy, and therefore you will accumulate energy.
Yeah, yeah. What about the effect on energy expenditure? I wanted to ask you about that.
Yes. This is for energy expenditure, the endocannabinoid system is also in the same line. That if you block the receptor, you have more energy expenditure, and so on and so on. However, probably in terms of this safety systems, there is a little bit of contradiction in terms of motivation for physical exercise. We published here that the absence of CB1 receptor is decreasing the amount of voluntary physical exercise in mice. And this seems to depend on the motivation. However, this could be, for example, linked. It's Francis Schauloff in my lab that is working on that, and he has this idea that he made a very nice experiments in which he put animals in a cage in which he says that this is a closed cycle in which the animals live for 15 days or something like that, and they have to work for food or for a wheel. Know that mice really love going on the wheel. So the animal has to work for one or the other thing. And so and this is very interesting because it is anorexia nervosa, for example, is if I ask anybody what is anorexia, I tell you it is less eating. Of course, you eat less. That's true. But one thing that they do also is to
compulsively exercise.
Impulsively exercise. So the idea to study the motivation for these two, we didn't finish the experiments. We don't know yet, but what CB1 is doing there. But we are working on that, trying to understand, to figure out what are the signals.
But I think if we abstract here, something that's important is the CB1 receptor seems to be generally important for motivation and sensitivity to rewards of any kind.
Yeah. That's
probably why the rimonabant.
And it's coming to another thing that I wanted to say. Coming back to what is an exostatic system. And when we say exostatic system, it is what we have to think immediately, and what you do rightly is future. So I spend energy now for something that maybe will be useful in the future. And for this, we need motivation. So an exostatic system is contributing to motivation. Okay? But it's not only for food. It's for many rewards, probably.
Right.
If you think about sexual interactions, they must be based on exostatic situation. Because you spend a lot of energy for something that maybe will be there 9 months later, which
it's a low probability of success for most organisms. Most organisms in the wild get very, very few opportunities to mate, so they have to make sure to take advantage of those opportunities when they arise.
Exactly, and they have to be a very strong motivation for that. So in that case, the balance between endostatic and exostatic, in my opinion, is very much towards exostatic. You have probably some endogenous things, but it must be, and that's why sexual relationship in all animals are starting to be the most visual, or the most olfactory, the most all the stimuli that
they're
are
so strongly sensory driven because they're exostatically regulated.
Most of it is exo.
Hmm.
So if you think of birds or stuff like that, is or insects even.
Yeah.
Uh, um, the stimulation is not the presence of a partner that is making, but is what it looks like, what it smells like, what is the noise that it does, and so on and so on. So it's like the cake of your grandmother. I mean, it's and yeah, and then one could apply this also to, but this is a little bit, it's for sure much more, uh, um, let's say, how you say, theoretical and difficult to prove. But one thing that I remember when I was a kid, and my father explained me Darwin and the evolution and uh, and there was this thing that he told me, okay, you animals and humans do things because this is favoring their survival, and therefore who does that survive and who doesn't do that doesn't survive, and therefore there is a selection. Who has the genes to do that will survive, and who doesn't has the gene to do that will not survive. I said, perfect. But that implies that what we do is useful. Otherwise, wouldn't have been selected. And so I started to ask him, sorry, but what is the use of a football match?
Hmm.
What is the use of a book? What is the use of a movie? What is the use of all these things that are uh, uh, not producing anything? They are not giving us food. They are not giving us. And then when Pierre we developed this idea of endostatic and exostatic, I thought, oh wow, could be that in humans this idea, this possibility of imagining, of having a future image of what an image of what the future will be became at some point fundamental,
Yeah.
because we are weak animals. We don't have teeth. We don't have that. But at some point, we were for sure on the verge of being
Mhm.
extinguished.
But we're clever, and that
There is
cleverness probably
evidence.
depends on the ability to imagine things that are not immediately
Exactly.
present.
So at some point, it becomes something that is entering in this motivation cycle.
Yeah.
That more you do, more you do. You do and more you do, and this is justifying why you do. And who does it is appreciated. If you tell me a good story, even if it is not at all useful, doesn't give me any food, I like it. And I like you because you are a good thing. So this is becoming something that is super motivated in
Yeah.
the
Well, I
and
mean, yeah,
of course much more
yeah.
difficult to
But
show
I
and
mean,
to
yeah,
prove,
yeah.
but
But I mean, if you think about human mating styles, the human mating system is basically one big externalization of, if you think about the way that human females choose males, they basically look to the group and say who is capturing people's attention, who is
Mhm.
winning the external game, and then that's sort of the selection criterion. It's very much based on external systems. It's an exostatic bias.
But there is also a selection based on the fact that somebody can tell nice stories,
Yeah.
that somebody can make up a nice. It's a good actor. So the fact that the actor, even if it is ugly, the actor is telling us the possibility that this person could be another person
Mhm.
is rewarding. But this is what I want to say. So for me, it is, and I see it very explainable by the fact that it is an exostatic thing. It's something that is done for the future. So
Right.
it's energy that I'm spending now, and a lot of energy. Think about how much we pay basketball players
Hmm. Hmm.
or soccer players in Europe, and we spend so much energy for something that is just telling us a story. But there is this
It has
fight,
no
and
immediate
there's
survival
somebody is
value.
making a goal or making a basket.
Hmm.
And I I think it makes a lot of sense.
Yeah.
But
Yeah.
as I said, it's
Yeah.
hypothetical.
Yeah. But I mean, part of the story you've told us today, or this view of the endocannabinoid system, is a lot of things don't make sense if everything was to be endostatic, everything was to be
Yeah.
homeostatically regulated. But as soon as you have a time component and
Mhm.
uncertainty in the environment, and a survival need to prepare and survive through uncertainty, a
Absolutely.
lot of things start to make a lot of sense.
You got to the point. It's exactly what I meant. Yeah, yeah. And as I repeat, it's extremely clear in the food, and then it can be applied also to other things. As I said, also Pierre was mentioning in his book this possibility.
Yeah, yeah.
I
So
love it.
what types of things are you working on today in the lab related to endocannabinoid biology?
Today, now we have a couple of things. One is this thing of different cell types that can produce endocannabinoids. That's something that is pretty much exciting. And then non-related to endocannabinoids, but very interesting. I mean, it is related, but not directly. It is that it's a paper that is coming out right now, which we generated a tool that is able to stimulate the activity of mitochondria,
Hmm.
which is something that but I don't know. I should speak with somebody to know why. But mitochondria you can inhibit as you want. You can there are plenty of poisons that are killing mitochondria. But if you want to stimulate it, then it's very complicated. There are some drugs, and we have a lot of diseases that are due to the fact that mitochondria work less than normal. So and some of these diseases are neurodegenerative disorders like dementia or Alzheimer, stuff like that. And we know since long time that all neurodegenerative diseases have a mitochondrial component.
Mhm.
But one could say, is it causal? Is a consequence? Is it a epiphenomenon? Because it could be, of course, that your cells die and therefore your mitochondria do not work. So to make it short, we had this tool that can increase a little bit, a tiny bit, mitochondrial activity. And in two models of Alzheimer, one model of Alzheimer and a model of models, animal models, so we are not a clinical side, but another model of dementia. You make a memory test and you stimulate the mitochondrion with this tool just at the moment of learning, and what they couldn't learn anymore, they learn again.
Hmm.
That's very, very, very
Hmm.
surprising and exciting.
And you said earlier there's some work coming out where specific cell types, astrocytes in the brain, are sensing particular fatty acids in the diet.
Yeah, and it's not coming out. It's some early
Oh,
observations.
okay, okay, okay. So we have
No,
more
no,
to look
yeah.
forward to.
No, but yes, we have the idea that now in the astrocyte field is coming out that the astrocytes are not all the same.
Mhm.
And so there is this idea that there could be populations that are specialized. There was a very famous paper on Nature some 1 year or 2 years ago by Andrea Volterra in Switzerland. He showed that there is a kind of glutamatergic population of astrocyte
Mm.
that is using glutamate to regulate neuronal activity. So no, another thing that we are doing, and I'm very excited about is the social transmission of stress.
Mm.
Yes. Uh, so this again was by Jaideep Bains. He published a paper some years ago in which if you take an animal, two animals that are two mice that are together, be living together, and then you remove one and you shock, and then you put back with the other one, the observer will start to make what is called anogenital exploration. So we start to put the nose in the anogenital area of a shocked one, and then we'll get an endophenotype like if it was stressed. So we'll get stressed by the stressed, not by the stress, but the stressed friend. Okay? So this was a very nice paper they published in 2018. And then we met and we said, okay, he told me CB1 is involved in everything. Why we don't check if it is involved there? And it is. So we published last year that it is CB1 in mitochondria of astrocytes of olfactory bulb that determine the fact that the animals make the observation. And on top of that, another thing that we observed is that we know that stress, if I get a shock, I will be unable to remember certain things that are not related to stress. So stress is like something that is making you focus. So you say, you get a stress, so now you have to focus on what is stress related. All the rest is not important. So if I'm stressed and I study mathematics, I will forget. Okay, because it's not stress related. But if I'm stressed and I learn that there is something dangerous, this I will get because it's related to the stress. And so we know that direct stress will impair this type of memory, a memory that is non-stress related. And we found out that CB1 in astrocytes of olfactory bulb is necessary also for the acquired stress, the socially transmitted stress, to inhibit the memory. So also the one that is just smelling the stress, smelling from the genital area, will become amnesic. And this depends on CB1 in astrocytes and so on. This is really, really a nice story.
Are there any final thoughts you want to leave people with, or anything you want to reiterate from our discussion about the endocannabinoid system and what it's doing?
Yeah. Well, I mean, it's a classical question, why you should study endocannabinoid system. I mean, I think that still, even if there is many years of work behind, there are still many things to find. And what I said at the beginning, I would reiterate, but the fact that the endocannabinoid system can be really seen as a kind of key to enter complexity. So it's a way that is that there are others. I'm not saying, but because it's so wide, so wide, yes, so diffused. Actually, there is a friend of mine who says that in the brain there are three systems. One main excitatory system, glutamate, one main GABAergic system, inhibitory system, GABA, and one main modulatory system, endocannabinoids. And then there is all the rest. And so this fact that you can use a tool to study how local modulation is acting on brain and body processes is still an advantage of a system that I like very much.
All right. Well, Dr. Giovanni Marsicano, thank you very much for your time.
Yeah, thank you. It was a pleasure.
Visit mindandmatter.substack.com to find all of my content. Hey everyone, I want to take just a minute to tell you about one of my cool new affiliate partners called Seed Oil Scout. Seed Oil Scout has a really cool map feature that helps you find restaurants in your area where you can find seed oil-free options. They also have a really cool product scanner, so you can take out the app and use the camera functionality of your phone to scan products in grocery stores and see what's actually in these things. Do they contain seed oils? Do they contain other things that you want to know about that aren't necessarily on the box. This will help you find places to eat and find foods to eat that actually have what you want in them and help you avoid those that don't. So if you're looking to cut back on your seed oil consumption, or you just want to know more about what's in the foods on the shelves at the grocery stores, download Seed Oil Scout today. If you go through the link in the episode description, click into that and you'll see a link to the Seed Oil Scout app. If you go through that link on my support page, you'll be directly helping the podcast, and you'll be able to download this really cool, easy to use app that will help you find healthier food options. So go through the link in the episode description and download Seed Oil Scout today.